started, we want to get this material. I don't want to rush today, but we want to get as much of this done today as we can. Guys, so that we don't feel like we have to rush. This will be the last PowerPoint for the exam. We will make headway into the remainder of nitrogen. We've actually done a bunch of nitrogen, knowing how my tendency to run behind. There is some nitrogen that is extremely important that you as biochemistry students really have to have. And knowing my tendency to kind of drag things out, I actually pushed that earlier. If you're wondering why that weird urea cycle happened, that's something you have to system not come back up, that's obviously not on you, that's on us, and then everybody just has to get the full extra credit. I mean, this is pretty cool class. The second part of the extra credit is another story, but that's, like I said, we'll talk about that later. Let's talk about making lipids. We're going to focus on this because we've already seen how we break lipids down, so this really is our last big metabolic unit. Most of our conversation is actually going to be on the synthesis of fatty acids, triglycerides, and the common membrane lipids. That's where we're going to see the chemistry. There's some interesting regulation that goes on there too, but the first half of this, probably the part that we mostly cover today, is the chemistry of those lipids. There are some other lipids as well that we want to look at, as well as this process and transport. That gets more to the heart of what some of you derisively call the biology. That's in there, and it's important. We will look at that for some of the other pathways. For example, cholesterol synthesis, which is something we might talk about today, but probably will get referred to in the next lecture. It's a very complicated pathway, and there isn't really, to be honest, all that much interesting novel chemistry in it, but the regulation for that is just like over the top interesting. So early on, with the focus on fatty acids, synthesis of triglycerides, and synthesis of the common membrane phospholipids, we'll look at the pathways, and that's really what you should focus on. Not ignoring the regulation step, that is important, but there's a lot of really good chemistry in there. And then we'll talk about the things like the steroids and some aspects of the transport. There, the real focus, the thinking should be much more about regulation, because that's what's interesting. So that's what we're going to talk about now. Lipid metabolism, the biosynthesis, again, mostly fatty acid-derived molecules. Regulation, kind of a both. And then the transport, how these very hydrophobic molecules actually get moved around. So we can start by comparing and contrasting what we saw when we looked at fatty acid degradation. That was in beta oxidation, a pathway that does not appear on the fact sheet, but that you are responsible for. And in that case, you'll remember our goal is, our objective is first acylating this with a CoA. And then we went through a series of oxidation, hydration, over-oxidation, and cleavage, removing 2 carbons at each step, restoring the CoA so that we can continue, and so we just kind of put this over and over. Each time around, assuming we were starting with an even number of lipid, which most of our lipids are, let's see why today, we generated two carbon fragments, except for the last stage, of course, where we generated, single 2 carbon fragments, at the end we generated 2 carbon fragments in the last step of the beta oxidation. The key is that this all took place inside the mitochondria, so it was segregated away from the cytosol. Most of our fun chemistry today will happen in the cytosol. That will make kind of sense to you. And we also, along the way, collected electrons in NADH and FADH2. You should probably know how much for each step of this. These are separate than the ones that for the further oxidation of the citric acid cycle. So just in the same way that in glycolysis we were generating NADH and FADH, not FADH2, but NADH anyway. So do we in the degradation of fatty acids. The key, of course, is that these are inside of the mitochondria, so no special odd transport mechanisms for getting those reduced carriers across the membrane are required. That was all beta oxidation. That's kind of a summary of most of our last lecture. What are we going to do today? Today we are going to make fatty acids, so that in some ways will seem like the reverse of the process. But here again we will see another excellent application of one of the things that we've talked about all the way since the beginning of Biochemistry 1, or at least very early on, and have reiterated multiple times throughout this course. If you have a very energetically favorable pathway, which beta oxidation is, you are not just going to turn the whole thing around, right? So we saw this with glycolysis and gluconeogenesis. Some things might be similar, but certain key steps had to be done differently because they were so energetically favorable. So the same thing we'll see is true with fatty acid synthesis. Certain aspects of this might seem sort of familiar, but it's really key to remember that it's not just the same pathway running backwards, particularly with energy-dependent steps. The key here is that, of course, we're going to do this in the cytosol. This is very favorable. Nature kind of makes sense. We don't want to be in a situation where we are synthesizing fatty acids just to break them down. We have one or two metabolic states in which we are contending with, and we want to either be breaking things down or making them. Segregating these out into different types of compartments is an excellent way to regulate that without having to worry about complex allosteric modulators. So there's nothing really fructose 2,6-bisphosphate like this, right? We do one in one compartment, another in another compartment, and there is, of course, some interchange of material we'll see, but we really don't need to do much more complex than that. In fact, you'll see with the regulator it will actually kind of be almost maybe what you would have predicted. Another key difference, of course, NADPH is going to be our reduced electron acceptor. When we break things down, we collect electrons in NADH, also have ADH2, of course. When we synthesize these, this is usually our electron source. We've talked about why that is, so I won't repeat that. It turns out that the NADPHs that we generated in the PPP, the pentose phosphate pathway, are not adequate for the job. They're adequate for keeping your cells from getting too oxidized and doing some basic biosynthesis. But as you imagine, lipids have a lot of reduced carbon, which means we need a lot of electrons. The number of electrons we have is too short of that, so we're going to have to see another way of generating those remaining NADPHs so that we can actually snap together these molecules. Biggest difference of all, each time we went through beta oxidation, we chopped off, for most of this, yes, but we'll say it for all for now, 2 carbons, except the last step, of course, where we had 2 carbon fragments. This process of synthesis is going to involve sometimes a 2 carbon fragment, but more often than not a 3 carbon fragment. So even if you look at the first round of synthesizing a fatty acid, right, that first round where you have the smallest molecules, 2 plus 3. So 3 carbon molecule and 2 carbon molecule generate a 4 carbon molecule. So once again, the math doesn't seem to add up, but hopefully when you see the chemistry, you will finally see what to do with that way. Just to remind you, the crucial molecules that are going to come into play here are these thioesters. Acetyl-CoA has been the one that we've seen so far. But of course, we don't need any of this, right? The only really important part is here. The rest of this is spacer arm. This is so that it can be recognized by the protein. So far, our thioesters have pretty much been acetyl-CoA, but they don't have to be, right? We could attach anything to a sulfur there in a long carbon chain that would do the job just as well, and that's really critical. Remember why it's critical. Carboxylic acids resonate a lot, esters resonate a little, thioesters, because of the poor overlap of these orbitals and these orbitals, do not resonate at all, jamming all the electrons therefore into this alkyl group, making that a very high energy molecule. So we talked about that quite a bit in class. Keep in mind that is true for all thioesters, not just for something magical about the CoA thioester. We'll see other thioesters. Anyway, fatty acid synthesis is going to take place in 3 different phases. We'll look at all 3 of those phases here. The first is we're going to have to take our acetyl-CoA from the mitochondria, which is where it's going to be produced, and send it out into the cytosol. So when I told you that even though we were going to be working in the cytosol, there will be some mass transport between different compartments. This is really what I'm talking about. This is the preparatory phase. We have lots of acetyl-CoA in the mitochondria, and we need to send it out into the cytosol, because that's where the synthesis is actually going to take place. In the cytosol, we need to convert that acetyl-CoA into a more reactive molecule. And the more reactive molecule that we're going to convert it into is malonyl-CoA, this 3-carbon molecule here. So you can probably look at acetyl-CoA and imagine how that's done. Here's kind of our old parent acetyl-CoA. Here's a carbon dioxide. So yes, if you're having flashbacks of the previous exam, this is once again jamming the carbon dioxide onto something. That's kind of what the chemistry will be. And then we'll have the actual reaction where we will build the molecule 2 carbons at a time. So we're going to start with 3 carbon molecules and 1 2-carbon molecule, progressively add 2 carbons each time, but use this activated 3-carbon molecule as the ratio, so that's the malonyl-CoA. Again, you can think about this molecule really as consisting of 2 parts. Here's the acetyl-CoA part, so this is the part that's actually going to be incorporated into our growing fatty acid. And here's the carbon dioxide, which is really the activated group. We know that these electrons right here can easily be localized into this structure here if the carbon dioxide is lost. So this is another way of saying, calling malonyl-CoA, I should say, is just activated acetyl-CoA. We saw something similar before, right? This isn't new chemistry. This is not the same enzymes, just to be clear, not the same molecules, just to be clear, but we've already previously seen that oxaloacetate could function as activated pyruvate. Again, these electrons could be lost. In gluconeogenesis, we actually saw the electrons delocalizing all the way down onto the oxygen and then grabbing the functional groups. But the same idea. We could essentially activate a molecule, make it more reactive by adding the carbon dioxide onto it, and then allow the carbon dioxide to come flying right back off. That's the basic chemistry, so it's something similar to what we've seen. Before we do so, however, we need to get our acetyl-CoAs, right? So we need to get our acetyl-CoAs. There are a variety of sources of these acetyl-CoAs, although probably the most common acetyl-CoAs would be acetyl-CoAs that come from, say, carbohydrate metabolism. I say that because it would make absolutely no sense to derive those acetyl-CoAs from lipid metabolism, because you would not be making and synthesizing, breaking down and synthesizing lipids at the same time. We'll see the mechanism by that works. So we have to get those acetyl-CoAs out of the cytosol, but only if we are in an energy-rich state, right? So how do we make sure to only send the acetyl-CoAs out of the mitochondria into the cytosol when we are prepared to make lipids? And the answer is we do so in the form of citrate. Acetyl-CoA cannot leave the mitochondria, which is a good thing, because then we can have a functional citric acid cycle. But strictly speaking, once the citrate concentration gets extremely high, right, once we have so much citrate that it matches the Km of the citrate transporter, it will start to spill itself out into the cytosol. Now, if this doesn't sound new again, that's because absolutely correct. Because some of the citrate is actually going to turn down glycolysis. We already saw that citrate is a negative allosteric regulator, right, of that substrate cycle. And that's because when the citrate concentration grows so high, it can actually go through the transporter. It's probably a good idea to stop the whole glycolysis thing. You really don't need any more energy. We're in a very energy-rich state. So the acetyl-CoA is going to leave the mitochondria in the form of citrate, and once it gets into the cytosol, of course, we need to break that apart. So we need to break that apart. We do so here with the citrate lyase enzyme, which we'll look at the mechanism in a bit. And that will give us oxaloacetate, right? Plus our acetyl-CoA. The acetyl-CoA will go back and enter into the process of synthesis. So what we've basically done is run the citric acid cycle and run the citric acid cycle in the opposite direction. A couple of interesting features about this are this is an ATP-dependent enzyme. Breaking this actual molecule apart actually isn't that energy required. In fact, it's quite favorable, so it's not clear why we need ATP. But let's continue to follow this anyway, right? So we can kind of take a look at that. So now we have oxaloacetate out here. We don't want oxaloacetate out here. We want oxaloacetate back here because we would like to continue the citric acid cycle going, ideally. We don't want to deplete the citric acid cycle of oxaloacetate. So what can we do? Well, we've already seen that malate can go back and forth either way. So one option that we actually have would be to send the oxaloacetate back in the form of malate, send a pair of electrons onto the oxaloacetate, reduce it, that converts it into malate. And we already know that there's this malate alpha-ketoglutarate transporter. We have seen that so many times in different lectures. We could send the malate back. But there's a reason why we're not going to do that. We're going to do something else. And at first, this might seem kind of counterintuitive. We're going to take the oxaloacetate, and we are going to reduce it to malate. But instead of the malate preferentially going back in, we are actually going to oxidize it to pyruvate, and send the pyruvate back into the pyruvate transporter. And at first, this seems to not be such a sensible thing to do, right? This could happen, but this is preferentially going to happen instead. We're going to send things back into the pyruvate transporter after effectively oxidizing it and losing the carbon dioxide. And remember, attaching carbon dioxide, attaching carbons to things is kind of an important goal. So losing carbon doesn't seem like it would necessarily be a very effective thing to do. So the questions we have here, well, we already know how the acetyl-CoA gets out. The questions we have are, first of all, what's going on here with this enzyme, the citrate lyase? And probably more importantly, why is it better to oxidize, right, to reduce the oxaloacetate to malate, and then oxidatively decarboxylate it, collecting some of the electrons right back onto pyruvate? So those are kind of our two big questions. In terms of the citrate lyase, the reason for the ATP has absolutely nothing whatsoever to do with the lyase reaction and everything to do with the fact that you just can't add a CoA to things. So here's the citrate, ACLY, this kind of represents the whole enzyme. What we're actually going to do is just add a phosphate group to that as the leaving group, and then the CoA can come and displace that. So in terms of energy usage, it turns out that the lyase step is just a reverse aldol reaction. You can see it happening here. That's what's breaking us back into oxaloacetate into acetyl-CoA. The actual energy usage of this is not, therefore, to break the molecule into two parts. We actually have to expend energy to restore the CoA. And in this case, phosphate is good enough. We have the good leaving group, so when the CoA comes in, it can displace the phosphate. And that's how we are restoring our CoA to the molecule, because we need to have acetyl-CoA. So this is where the energy requirement comes from. Let's take a look at this other question now. Why in the world, instead of doing the straightforward thing of just sending the malate back in, do we instead reoxidize it, lose a carbon atom, send it in pyruvate, which again is going to take us some energy to build back up to oxaloacetate, right? So this is one of those anapyronic reactions we saw. We're going to have to convert the pyruvate back into oxaloacetate, but that's going to require the reinvesting the carbon dioxide and taking some energy. Seems to not be the proper thing to do. Seems to be completely counterintuitive. It actually turns out to be really important, something called the reductive swap. Remember, I told you that the pentose phosphate pathway is not adequate enough to provide us the electrons that we need to do lipid synthesis. We're just going to come up short. I think it gives us actually about 60%. The 60% is not adequate. So where does the rest of it come from? Well, here's where it actually comes from. Look at the series of reactions we're doing. We are first going to reduce oxaloacetate to malate, right? So we're running in the reverse direction of here, because this is a nice slide, so I kept it. And then we're going to reoxidize the malate to pyruvate. But notice what we've done. We've essentially collected those electrons on NADH and net transferred those NADH electrons to NADPH. So we had NADH there, right? So we had some NADH. We transferred the electrons net to NADPH. And this is what's called the reductive swap. Rather than send it back in the reduced form, which really isn't necessary, this series of reactions here are moving electrons from NADH to NADPH, and this is where we're going to get the remainder of the electrons that we need for lipid metabolism. And since pretty much the only thing we're going to do with that acetyl-CoA and the cytosol is make lipids, this ensures that you're only generating extra NADPH when you need to make lipids. In other words, the pentose phosphate pathway is handling all the other demands, but when you need to step it up and make some lipids, you do the reductive swap. You essentially take electrons net off of the NADH and transfer them onto the NADPH. And that's the utility of sending this back in the form of pyruvate. Okay, so great. It's time to go and start using some of these electrons, start making some lipids, particularly fatty acids, which we're going to focus most of our time on. Here is the enzyme that's going to start this off, our preparatory step. electrons, so adding the carbon dioxide on is feasible. This is actually, you can see here, a multiple subunit enzyme with the biotin carrier protein is in the center. So once again, we have a biotin attached by a lysine residue, the long spacer on there, to the protein, right? So that's a lysine on the protein attached to the biotin. We know that we're going to have to use ATP to attach the carbon dioxide to the biotin. But I didn't tell you that that was kind of on the last test. And then the second major domain of this is going to carry out the transcarboxylase, where the carbon dioxide is removed from the biotin, attacks the acetyl-CoA, and that's how we're going to make the malonyl-CoA. So that's kind of the preview of the chemistry. This is actually right from the previous exam to show you again. You have the bicarbonate. The idea of using ATP is that this is just not sufficiently reactive. So we need to have a good leaving group to it. And then the CoA can come in and attack that biotin, the carboxy biotin, and that's how we actually start the process of adding the carbon dioxide and generating the malonyl-CoA. Really critical that you are only doing this if you absolutely have to, right? So unlike other biomolecules we've seen, malonyl-CoA is pretty much only going to be useful for making our fatty acids, or doing that sort of lipid synthesis. So this is pretty much a committed step. Going through to making the malonyl-CoA is absolutely committed. And so as a result, there are multiple modes of regulation that actually involve these. But not surprisingly, the most common modes, the universal modes, are actually phosphorylation and dephosphorylation of the carboxylase. The kinase, that's going to add the phosphate group, that's that AMP activated protein kinase, not protein kinase A. Right? This is the AMP activated kinase. This is the signal of low energy status. Remember, this is the one that gets activated by AMP when your ATP stores have been depleted. If your ATP stores have been depleted, there are a lot of really good reasons not to be investing energy into making lipids, so they'll actually just phosphorylate it. Protein phosphatase will strip that phosphate group off, and so that will get us restored to the state. And so this is the major mode of regulation to ensure that at low energy situations, you are not devoting precious ATP, nor for that matter citrate, right, into actually starting to try to make lipids. There's also an allosteric regulatory mechanism, and here again, they're actually kind of the regulators that you might imagine. If you have lots of citrate, this is being sped up. If you have lots of palmitoyl-CoA, long-chain fatty acid, it's being slowed down. But I want to show you the mechanism, because this is a kind of a mechanism of regulation we haven't seen before. These are allosteric regulators, but they're not working by just activating or increasing Km or Vmax in the logical way like every enzyme you've seen. They're actually changing the polymerization, or you could say the quaternary structure of the protein. So as it turns out, the carboxylase is only active when it's a polymerized molecule. So each of these purples is meant to represent a single individual carboxylase. And in fact, only when they're in this long filamentous form are they active. So this is an actual electron microscopic image of the carboxylase, and it forms these long polymers that can be seen with the electron microscope. So there are a lot of these here. And when these are broken up into monomers, the protein or the enzyme becomes inactive. It actually doesn't function. So who stabilizes the polymer? It turns out that citrate binds in such a way as to stabilize the polymer. You have a lot of citrate, you want in the cytosol, you want to make fatty acids, the polymer becomes stabilized by binding to the citrate. Who breaks up the polymer into monomers? Palmitoyl-CoA, long-chain fatty acid, binds to it, causes those polymerized subunits to dissociate. So this is a mechanism like none we've seen before. This is a regulatory mechanism at the allosteric level that's determining whether or not the carboxylase is on. Because again, you don't want to make this malonyl-CoA if you don't have to, right? If you're not going to make lipids, do not make malonyl-CoA. Interestingly enough, the only other thing that I wanted to tell you on this slide, which again, another important mode of regulation, is that this malonyl-CoA is not only used to make lipids, but it is also a negative allosteric regulator of the carnitine acetyltransferase. You may recall back from our previous lecture, the carnitine acetyltransferase is the enzyme that starts the process of attaching our molecules onto the carnitine, right? So the fatty acids onto the carnitine so that they can move across. So this molecule, malonyl-CoA, is actually inhibiting that process. which kind of makes sense. You do not want to be making fatty acids just to ship them off to get degraded by beta oxidation. So the malonyl-CoA, if it's present, that carnitine acetyltransferase is inactive, so those freshly made lipids don't immediately get delivered to the mitochondria and chopped back up into acetyl-CoAs, because that would really be a fuel cycle. Okay, time to make these fatty acids. It turns out that the mechanism by which this works is absolutely evolutionarily conserved, all the way back from bacteria, all the way to us. The structures of these enzymes are also highly conserved. The only thing that really differs is the domain structure of these, the domain and subunit structure. So in the case of bacteria and also many plants, all of these different enzymes are actually 7 different polypeptides. By yeast, they have fused them genetically into just 2. And for us, they're actually all found on 1 polypeptide chain. So each one of these 2-letter codes represents, except for the one in the middle, yes, represents a different enzyme functionality. You are not responsible for memorizing most of them. There are 2 that I guess you would kind of need to know, but those 2 should be kind of obvious as we're going along. You do need to know the whole pathway, but worrying about the exact 2-letter code for which enzyme carries out which particular step in that pathway, that's kind of not the important point, right? You should actually see how those lipids actually get made, okay? So here's the different domain structure and subunit structure, but again, the enzyme structures themselves are all conserved, as is the mechanism, so this is kind of true across all of evolutionary history. Really important, sitting right at the center, is this acyl carrier protein. The acyl carrier protein has one important cofactor, lysine linked phosphopamphine group that has sulfur at the end. So much like coenzyme A, it can form thioesters. And in fact, quite often what we're going to see is the conversion of one thioester to another. So for example, we will bring in an acetyl-CoA and an ACP. That's again that one right in the center of all things, right? That's where it's physically located. And form acetyl-ACP. So just add 2 carbons to the ACP and lose the CoA. And we can do the same thing you'll see with the malonyl group, bring in an ACP and get malonyl ACP. Chemically, the 2-carbon molecule or 3-carbon molecule doesn't know that anything has changed. So this just happens essentially as an equilibrium. If there's more acetyl-CoA, it loads up the ACP. If there's more malonyl-CoA, it loads up the ACP. It's essentially isoenergetic, so no exchange or other ligand groups are required. And chemically, it's a thioester being exchanged for another thioester. Okay, before we look at what's happening, because it looks pretty complicated, once you go through it a couple of times, you go over the whole thing. But instead of bringing in all the different enzymes and all the different subunits at once, let's just strip the chemistry down to its most minimalistic state. Start the process here on this slide to see how the chemistry is working, and then we'll go back and we'll see how we actually build an actual fatty acid. Because I think it's useful before we start bringing in all these other domains to just look at the basic chemistry. So what do we have? Well, we're going to be doing a nucleophilic reaction, so we need a nucleophile, we need an electrophile, and we need a leaving group. And for once we have all of those things, how rare is it in this class that we really have everything that we need, right? Here is going to be our nucleophile. That's actually going to be the carbon here of malonyl-CoA. Here's going to be our electrophile. That's going to be the carbonyl group here. We know that this carbon dioxide is leaving, and that's going to be what delocalizes some electrons onto here. And so these electrons on the CH2 group are going to attack this carbonyl, and that's how the carbon-carbon bond is going to get formed, right? So we lose the carbon dioxide group. Again, it's not happening by itself. The enzyme is present there. We're losing the carbon dioxide group. That's going to put a pair of electrons here, or a double bond doing the attack. It doesn't really matter how you want to perceive that. That makes a carbon-carbon bond, and notice we've now extended these 2 carbons, 3 carbons, lost 1, so there's 2 carbons into this 4-carbon molecule. You'll notice here, right, that the malonyl is on the ACP, the acetyl-CoA is on another one of those domains. That's the KS, the chemotherapeutic isomerase domain, but don't worry about why that's called that. Just KS, it's fine. But we have not finished yet, because you notice that we have a pair of carbonyl groups, and you know that we don't have just random carbonyl groups coming out of our fatty acids. So we got to get rid of those carbonyl groups, right? We need to reduce those. And so how do we reduce things? Well, we have electrons. There's those NADPHs, which were generated either in pentose phosphate or just recently generated in a reductive swap. We reduce them right to the alcohol. We lose a water 2 plus 3 is equal to 4, we of course lost that one carbon from the carbon dioxide. That was the CO from the malonyl-CoA. That's the chemistry. So everything else you do, as we build out this bigger, progressively bigger and bigger and larger lipid, is just a repeat of this chemistry. In the same way we keep repeating the chemistry in beta oxidation, we are repeating the chemistry here. So let's bring in a little bit more detail before we start to build out the whole lipid. Now I'm starting to show you the two relevant domains. Here's the KS domain, here's the ACP. So you can start to see how this works spatially. You'll see why we're doing this step by step when I actually show you the enzyme. It's a bit more complicated looking. We have our malonyl-CoA attached to the ACP. We have our acetyl-CoA, which is only the time we're going to use acetyl-CoAs in the first step. We lose the carbon dioxide enzyme catalyzed. A pair of electrons here attacks that carbon here, and now everything is on the ACP, notice. So here were the first two carbons attached to the ACP. Here they are here. Notice that we added these next two carbons, right? At this point now, another domain comes in, and we do our first reduction. Don't worry about what domain that is, we lose that name. Just remember that NADPH is doing the reduction. Then we're going to lose a water. That another domain is going to catalyze the loss of the water. And then another domain, and then now we have our 4-carbon molecule. So this would be considered one pass. Of course, we're not making 4-carbon lipids, right? We're making much larger lipids. So we're going to have to repeat this several times. So now let's kind of bring in the whole enzyme. You could hopefully see why I wanted to start with some of these smaller pictures of it. Here we go. These are each of the domains in their rough spatial approximation. The only domains that you need to know by their two-letter codes are what's happening on the KS and the ACP. But just keep in mind, it's not like NADPH is just floating by, or a water molecule is just going away, right? There are active domains that are close to the proximity of the active site where the substrate is being held that are catalyzing these reactions. But the only critical ones to know are the ACP and the KS. So let's take a look at what's actually happening here. We're going to start our first round, and our first round only, the only time in this entire process where we directly use an acetyl-CoA is going to be to install those first two carbons. So we start, essentially, by adding a CoA initially to the ACP, and then that gets transferred onto the KS domain. So the root is acetyl-CoA to ACP, right, domain, to the KS domain. That's what this represents, just to be clear. This sulfur is not just hanging out in space, it is part of the KS domain. So it's bioester, so we went on acetyl-CoA to ACP, and then ACP moved it over to the KS domain. So now we have the two carbons attached. Of course, we need more carbons, right? We need additional carbons. So now we're going to transfer malonyl-CoA to the ACP domain. It's not going anywhere. It's on the ACP domain, right? The ACP domain, the KS domain is already full. Now we're going to do our nucleophilic attack. We're going to lose our carbon dioxide, putting a pair of electrons here. They're going to attack the carbonyl group. In this case, the whole protein complex is going to be our leaving group from this step, right? So our whole protein is going to leave, and you'll see that we've just extended ourselves out now to 4 carbons. 1, 2, 3, 4 carbons, all on the ACP domain, right? Other domains, again, which you do not need to worry about what their names are now going to catalyze our reduction. We're going to reduce our carbonyl group, right? From our carbonyl group to an alcohol group. Not this one. If you're asked to draw this out, please reduce the right one. It won't make any sense if the wrong one gets reduced. Really common mistake when this is drawn. This is why I'm going through this in such slow, painful detail. Giving us a double bond with the loss of water. Right? So the double bond with the loss of water. And one more reduction. So here's our second NADPH group. And now we have our 4-carbon molecule. Of course, we don't want to just have a 4-carbon molecule. Our goal here is to not make just purely a 4-carbon molecule. Our goal here is to make a much larger molecule. So how are we going to make a much larger 4-carbon molecule? We need to transfer our 4-carbon molecule from the ACP domain back onto the KS domain. So that's the last thing that this round is going to catalyze, is to move our 4-carbon molecule from the ACP domain onto to the Ks domain, again, isoenergetic, so there's no energy required. The question is, well, why are we going to do that? The answer is we need to add more carbon. And of course, to add more carbon, how are we going to add more carbon? We're going to add more carbon by putting now a malonyl-CoA on the ACP group, and once again doing the attack chemistry and going through the whole process again. So notice we did the attack here, and we just added now, we've gone from 4 carbons and 2 carbons. Now we've made our 6-carbon molecule, right? We just added 2 more carbons. And this cycle will continue in each round. At this point forward, after the first round, every single time after the first round, it's going to be adding a malonyl-CoA. In the first round, we start with a malonyl-CoA and an acetyl-CoA. Every subsequent round of this chemistry is using a malonyl-CoA. And of course, we just continue to add carbons and carbons and carbons. So this is our first, right, acetyl-CoA. We're pushing it all the way out here, and then this is the palmitate molecule that we are trying to make. Okay, and that's it kind of for the chemistry of the beta oxidation. There's more, but just to kind of summarize where we are so far, here's a nice comparison of what happened in beta oxidation and what happened in fatty acid synthesis. Remember beta oxidation in the mitochondrial matrix. This happened in the cytosol. Our carriers were the CoA in beta oxidation. Our carriers were the phosphopanthine, but the same sort of chemistry, both in bioesters. In beta oxidation, we used FAD and NAD. And in fatty acid synthesis, it was always an ADPH. And in beta oxidation, we had acetyl-CoA. In fatty acid synthesis, in the first round, anyway, we had an acetyl-CoA and a malonyl-CoA. And then in every single subsequent round, we're just adding malonyl-CoAs. Couple things, just in terms of the regulation that now, once again, might start to make a little bit of sense. These genes are, of course, not only regulated allosterically, but also if you are in a very energy-rich state, energy-rich tissues, upregulating the gene expression. So coming back to something we talked about before, the cellulose 5-phosphate. Remember, that was something that actually came out of the pentose phosphate pathway. One which, of course, is going to be very active if you are generating lipids. You're going to have a lot of pentose phosphate going. Even with the reductive swap, about 60% of your NADPH is actually coming from pentose phosphate, which means you're building up a lot of xylose 5-phosphate in those cells that are making lots of lipids. So it turns out that the xylose 5-phosphate, in addition to just being a 5-carbon sugar going through the process, a small amount of it's going to get siphoned off and used as an allosteric regulator. It's actually going to activate the protein phosphatase 2A. The reason it's going to do that is that it's going to remove phosphate groups from this transcription factor here, SHREB. Poorly named, but it is what it is. TREB is actually going to go into the nucleus, and the genes that it's going to transcribe are genes that are involved in lipid synthesis. And again, this makes sense, right? You are in a situation where you have lots of pentose phosphate running, you have lots of xylose 5-phosphate running. That act present, that probably means you're doing a lot of lipid synthesis, and so you can take the burden off of the existing enzymes by transcribing more of them. So this is another connection between all of those pathways. And yeah, that's it. Or, you probably figured out that it's not, right? Because this is not necessarily the largest lipid you need to make. It is, on the other hand, the largest lipid that this particular enzyme complex can make. But it turns out that there are cases where you need 18, 20, 22s, and 24s. Those are made by enzymes that do the exact same chemistry, they just have a bigger active site, right? For making anything longer, you need a larger active site, but all of those domains can be replaced. So we're not going to go through the chemistry again, because guess what? The chemistry is exactly the same. But instead of being liberated as a free fatty acid, if you need to make longer lipids, they're transferred to these elongase enzyme complexes, and that will just add a couple of extra carbons to them for your C20s, your C22s, your C24s. You don't make much, just to be clear. Those are fairly rare lipids. But if they are needed, they are made using the exact same chemistry, just using a complex of enzymes that has a larger active site so that all of those different functionalities for all the different active sites can continually reach the lipid that you're trying to build. So those are called the elongase complex enzymes. What about unsaturations? It might seem that, well, we already had an unsaturation, so the logical chemistry would be just stop where you need the unsaturation, right? If you had a double bond, we could save ourselves some electrons by not getting rid of that double bond. It turns out that that would have been great if nature did that, but much like the ribosome is pretty much an unintelligent machine, it will just make anything that you feed into it as long as it involves amino acids. The fatty acid synthase doesn't know anything. It has no programmed instructions. As long as acetyl and aldehyl-CoAs are coming in, and as long as the active sites can reach the particular functionalities, they'll just keep going. And what that means is that even if you need to have an unsaturation, you want to make a particularly unsaturated fatty acid, you're going to have to do that later. That does not happen on the fatty acid synthase complex. Instead, there are actual desaturations. The consequences of this are actually quite significant, including for our health. The most obvious of these is that you cannot have an unsaturation in any specific position that you want. You can only have unsaturations in positions that nature has evolved desaturates for. And the most common unsaturation is this one here in the delta 9 position. So that's the one that we're going to look at. And just to be clear, I don't know, if there's some reason to have, it would be great for an organism to have delta 8, it doesn't matter because there's not a desaturase to make that thing. So unless that would have come about maybe by mutating this, you can't have it, right? So it's not going to happen on that actual fatty acid synthase complex. Okay, so anyway, what's the big deal about this? This is actually kind of a really interesting enzyme. Because when I first started reading about this, I thought there must have been a mistake in the textbook. I mean, I must have found an error in the textbook. I got really excited, right? Because, wow, no, something's wrong backwards. And let me show you what the mistake would seem to be. We need to oxidize this carbon-carbon bond, right? That's the goal here of this chemistry, to oxidize this carbon-carbon bond. And what will your textbook and my notes and any textbook that you say is the absolute necessary cofactor for oxidizing this bond? The answer is NADPH. And that just sounds wrong. Maybe if they said NADP, I would have been like, yeah, okay, that makes sense. I mean, because NADP is good at scavenging electrons, right? It's could be used as an oxidizing agent. Not its most common application, but maybe this is just an exception. But it's not the case. The actual oxidizing cofactor for the oxidation is NADPH, which, by the way, it is not the case that, in this case, that it can all of a sudden accept 4 electrons, right? Instead, it's actually kind of interesting because from the molecular or the chemistry perspective, the point of this enzyme is not to oxidize this double bond at all. This is actually a very, very, very strange electron transport complex. And just to be clear, it's not the same electron transport complex, right? There's no complex 1 and 3 and 4 and ATP synthase, none of that. But it is a version of an electron transport complex. And what was the oxidizing agent in the electron transport complex? That was oxygen. And so the purpose of this enzyme from a chemical standpoint, not from our biological standpoint what we want, but from a chemical standpoint, is to convert oxygen to water. And so the question is, what did we do in the electron transport complex to make oxygen into water? We fed it NADH. What is the problem with, what is the source of the electrons here? NADPH. So these electrons, NADPH, are not being used to somehow introduce a double bond. They are being used, right, to reduce the oxygen to water. What was the stoichiometry problem that we had in complex 1? The stoichiometry problem that we had in complex 1, back in the electron transport complex, was that NADH does not provide enough electrons to generate the water. You needed to have 4 electrons. NADH has only 2. What is the stoichiometry issue of this NADPH reducing this oxygen to water? There's not enough electrons. In our electron transport complex, that was accommodated by bringing in a second NADH. In the desaturase enzymes, the second set of electrons comes from the lipid. The lipid oxidizes those electrons and then passes those electrons onto the water. And that's why NADPH is the key cofactor for the desaturase enzymes. The flow of the electrons is NADPH to oxygen to make water, a task that cannot be completed without another pair of electrons, which actually come from the oxidation of the lipid. And that's why this is the cofactor. This is how a reducing agent somehow gets dragged in, right, to oxidizing a carbon-carbon bond on a long aliphatic chain. So that's the chemistry of that. In terms of some of the other necessary desaturations, there are other ones that you can introduce, but there is a problem in human or animal physiology in general. You need to have all of these fatty acids, or at least they are considered to be nutrients. Some of these I'm sure are pretty common. It's the omega-3s that you are taking pills for. Why are you taking those in tablet form instead of making those? It turns out that this reaction here, the conversion from 18:1 delta 9 to the introduction to delta 12, can only be done by plants, which means even though you need all of these other fatty acids, including some down here that you could synthesize, this process here represents a bottleneck. You have to eat these from plants. And so if you're wondering, no, wait, I'm eating these from fish. Fish are not plants. There's not that much bio in this class, Dr. Astro. But I know a fish is not a plant. Yes, but the fish or is eating a fish which has eaten some algae, which is kind of a plant for our biochemical purposes. And so this is really the bottleneck. All these other lipids are things that you need to have, but you cannot endogenously synthesize them, so they are in fact essential lipids. Okay, that's it for fatty acids. Let's make something out of fatty acids now. The common precursor of the two major molecules that we are going to make is phosphatidyl. Phosphatidyl is the common precursor for the triacylglycerols, how you're going to store it, and for your membrane lipids, or at least your membrane phospholipids. So we should see where the phosphatidyl comes from. Really two different paths. You could siphon some dihydroxyacetone phosphate out. That's possible. Glycerol also, this is a case perhaps if you've just converted pieces, literally individual pieces of triacylglycerol. Together there are these two different pathways, one which requires NADH, the other which requires an investment of ATP to activate, right, the glycerol into the glycerol phosphate. Then you need an additional investment of energy for the attachment purposes, but again, that's not a surprise. You know exactly what's happening here. We need to make a better leaving group, so for each one of these we need to attach to CoA. Then in this case, this requires the AMP leaving group. And then we just have the nucleophilic attack of these hydroxyl groups on the amylase of fatty acids, and this gives us the phosphatidic acid here. So this molecule here, this is our common intermediate for making either triacylglycerols or the membrane phospholipids, kind of whichever of the two of those things you need most of. In terms of making the triacylglycerides, which you now realize requires a little bit of energy to make, the last thing you need to do is to actually take off this phosphate group, bring in another one of your activated fatty acids, and now you have the triacylglycerol, and those, of course, are stored in these lipid droplets, primarily inside of the adipocytes. So this is where triacylglycerides come from. So you just went through this process and stored them. Last major point, therefore, to kind of add to this before we look at the membrane lipids, this explains why, an eternal question, if you consume lots of carbohydrates, you gain a substantial amount of body weight in the form of fat, right? So that's kind of the end of this question. Remember, lots of carbohydrates means lots of citric acid cycle, means lots of citrate, so much citrate, in fact, if you've had a lot of carbohydrates, then it pours into the cytosol, you get those fatty acids, they become acylated, attached up here to the glycerol, and this is where we get the triacylglycerides. So all of those excess calories from the carbohydrates are ending up being stored in the form of fats, and this is the chemistry that explains why. What about attaching or making actual membrane lipids, the most common of which are the phospholipids? So those are the ones that we'll spend most of our time actually looking at. You will recall back from our biochemistry 1 discussion that despite the picture you have in a bio textbook where the phospholipid membrane is just some big phosphate head group and some lipids here, there are actually a variety of different head groups forming different types of lipids. These, of course, carry different charges at neutral pH, they carry different structures, they carry different shapes, and so different lipids, different organelles, I should say, will have different requirements for these various different lipids. We've already seen, for example, the importance of cardiolipin in holding together complex 1, 3, and 4. So you need to make these on demand, but again, they're not made for the same amounts in each organelle, but rather distributed to the various organelles that require one or specific types of lipids. There are in general two different strategies, therefore, by which we can convert our phosphatidate into one of these membrane lipids. Bacteria and eukaryotes use strategy 1. Only eukaryotes use strategy 2. I have a couple of examples after I show you the basic strategies here. I'm not going to show you all of them, but take a look at them. You'll see what I mean. I included a couple of extra slides that just show you some of the common lipids and how they can be accomplished with one or both of the strategies. But let's just kind of look at the basics of the strategy, what they're going to carry out. We know from every single molecule we've talked about in this course in Biochemistry 1 that if you want to connect two pieces together, that is an energetically unfavorable reaction. So biosynthesis in general is thermodynamically unfavorable, and that's going to necessitate a leaving group. So we saw that in the case of proteins, the leaving group was additionally the ADP group, swapped out with the tRNA, right? We saw with carbohydrates that leaving group was the UDP leaving group, right? So it turns out that for the synthesis of lipids, the leaving group in question is actually going to be C, derived from CTP. So it's as though nature has somehow segregated out, if you will, each one of these different nucleotides to have a different function. GTP, mostly for timing. UTP, mostly for making carbohydrate glycosidic bonds. CTP, mostly for the assembly of membrane phospholipids. Of course, they all end up in DNA or RNA, but this is kind of their metabolic role. Anyway, so our goal here is going to be to make different leaving groups. And what differs in terms of these strategies is who gets the leaving group, right? We have all these nucleophiles around, all these potential electrophiles. The leaving groups, in theory, could go onto the fatty acids, or the leaving groups could go onto the head groups, right, which you're attaching. Both of those things are possible. So here's the first strategy. You can see that the goal is actually going to be to make the diacylglycerol activated with the CDP group. So here's our leaving group. And then bring in a hydroxyl group from the head group. And then this is how the head group is going to be attached to our phospholipid. So we have the phosphatidate. We're attaching the CDP group to it, right? So this was originally phosphatidate. We've attached CDP group to it. We have a head group that has a hydroxyl group in it. And here we go. This is strategy number 1. Found both in bacteria and in eukaryotes. Now, eukaryote-only strategy, the one that you will only see, is when you actually can activate the head group with CDP and use the hydroxyl group on the diacylglycerol to do the attack, and this is where you get the membrane phospholipids. So here are your two different strategies. What they both have in common is the use of the CDP group, originally derived from CTP. Everybody can do that first strategy, only eukaryotes can do the second strategy. So again, if we're looking at something like phosphatidate, we want to first convert that into the CDP diacylglycerol, so that takes CTP. Once again, for the, I don't know how many times in this course, the reaction is driven forward, by the loss of the pyrophosphate group, which gets destroyed. And then look at this. You have all of these possible head groups, and what do they all have in common? They all have a hydroxyl group. You have serine, you have molamine, choline, glycerol, right, inositol. They're not all done by a single enzyme, but rather specific enzymes will come in and convert the phosphatidate into the CDP diacylglycerol, and then have the serine attack and displace the CDP group. And so, not, I guess, the best of slides to kind of see here, which you can see examples of this particular strategy laid out for you, right? So first activate, then bring in a hydroxyl-containing group, displace it. Occasionally, additional chemistry will happen after that, so methyl groups can get added, oxidations or reductions can happen to modify those head groups, but that's the basic strategy that's actually there. This illustrates here an example of the other strategy, in which case you can see that we're actually going to add the group to the head group, and then do the attack right onto the pre-existing glycerol. Just take a look at these. There's not really a lot of point in going through each one step by step right now. Better to go take a look at these at home and try to get a sense of how each one of these different types of phospholipids are built. Again, not for the memorization purposes, but just to understand metabolically speaking what are these two different strategies. Second major component, therefore, that's actually actually present are the sphingolipids, which have a unique biosynthetic pathway. Once again, using some of the principles that we've already seen before. Key here, you're not memorizing this pathway. Unlike, say, the pathway for fatty acid biosynthesis, which like you really should know, I just want to point out some of the key chemical features that come into play here. And those are what's really important, right? So it's not the actual memorization of the entire pathway. So that's kind of my pre-existing warning. Just to remind you, however, back in biochemistry 1, when we put these sphingolipids together, we always kind of started with the sphingosine alcohol. You'll remember that we acylated on here by a peptide-like bond on one fatty acid chain, and the head group went on here. I think what I'm trying to emphasize is that this is kind of how we categorize this in biochemistry 1. This is not at all how sphingolipids are actually made. In fact, sphingolipids are actually made by first building in that long carbon piece, which turns out to come also from a fatty acid. So you know that fatty acid that we just made, that we could have, in theory, converted to store it, or put it into a phospholipid. Another possible fate for that is actually just to use it as the long carbon chain for the actual sphingolipid. So its origin, the long carbon chain, is actually derived from the fatty acid. It won't be for long because of all this chemistry we're actually going to do to it. The other second of 3 really significant things here is that where's the rest of the molecule actually going to come from, right? So we saw the long carbon chain. Turns out the rest of this molecule is going to come from an amino acid, and that amino acid is actually serine. So we're going to add the serine with the loss of the carbon dioxide. That's going to be a PLP-dependent process. Remember, the carbon dioxide has to come off. That will allow us to stabilize, generate a resonance stabilized formaldehyde. And you can see here now we're starting to build up the sphingosine molecule. There are other modifications, the addition, for example, of fatty acyl-CoA onto that nitrogen, as well as the introduction of the double bond using an enzyme complex similar to what we saw. And then the addition of the head groups itself is actually very similar to what we saw when we looked at the phospholipids. So key features here, I guess if you're wondering, okay, so what should I know, Dr. Petro? Good question. The origin of the long carbon chain, that's actually coming from a palmitoyl-CoA, so that came right out of our fatty acid synthesis. The origin of the rest of the molecule, that's from the amino acid serine, right? So serine actually comes in, and it's going to lose its carboxyl group, but it's going to become buried here. This part of the molecule is generated from serine. Origin of the double bond, you've already seen these desaturases and kind of how they work, so it's not really anything new to add. But those are really the key features to understand about how sphingolipids get synthesized. Another really important class of lipids has nothing to do, well, not directly with membranes anyway, but these are actually the signaling lipids. So remember, we talked a lot about signaling this semester, but one of the very common signaling molecules that we discussed a little bit in Biochem 1, but also in Biochem 2, were these icosanoid molecules. And again, remember that these are things that were involved largely in things like blood vessel constriction, platelet aggregation, airway constriction during asthma, and those are also actually going to be derived by fatty acids. So we're not going to look again at the entirety of the pathways for these things. They're quite complex, and a lot of the details aren't necessarily the most important thing. The significance is that they're all going to start with this molecule we talked about last semester a little bit, this omega-6 fatty acid called arachidonic acid. And there really are 2 different possible pathways in which these can move. One of these pathways, you can see upwards here, are to make a family of molecules that are known as the leukotrienes. These are catalyzed by enzymes called lipoxygenases. We'll see what the lipoxygenases do shortly. The second possible pathway, again for pro-inflammatory molecules, are to synthesize the prostaglandins. Here's one example of these. And these are going to make molecules like prostacyclin, lots of other prostaglandins, thromboxanes that's involved in blood vessel constriction, as well as the platelet aggregation. Key feature, this pathway here, the cyclooxygenase pathway, are inhibited by cyclooxygenase inhibitors. There are 2 types of the cyclooxygenase enzyme, COX-1 and COX-2. These are the targets of your non-steroidal anti-inflammatory drugs. These are your targets of your aspirin. These are the targets of your ibuprofen. They inhibit things like headaches and inflammation by blocking this pathway. So headaches, for example, will come about because of constriction of blood vessels. That is blocked, again, by these COX-1 and COX-2, these inhibitors of these enzymes. They do not have any impact whatsoever by these leukotriene molecules. These inhibitors of these enzymes do not block the lipoxygenases. They do block the cyclooxygenases. So what are the differences? Well, here are the cyclooxygenases. You can see here down in this row. They all involve the addition of oxygen and the formation of these ring structures. So you can see what some of them do, the platelet aggregation, for example, clotting. These are the additions of oxygens and the formations of rings. So this is the pathway down here. You have the starting arachidonic acid. All of these molecules, in one form or another, will interact with the cell surface protein, quite often a G protein coupled receptor, and the results of that activation will be inflammation that can all be blocked by treating inhibitors from one of these two. Again, it could be aspirin, which could be ibuprofen. Here are the leukotrienes. Again, starting with the same parental substrate, the omega-6 arachidonic acid, right? Here's what the lipoxygenases do. They generate these peroxide species, which are all called leukotriene molecules. These also promote inflammation. You'll notice the difference. They do not involve ring structures. In this case, both oxygens are present in the same part of the molecule. So these are the leukotriene molecules. These are, in fact, molecules that block their actions are popular drugs for the treatments of inflammatory conditions like asthma. For example, montelukast, you may have seen, or not, seen drug ads for, blocks leukotrienes from binding to their receptors on cells and prevents airway inflammation. So this is a common treatment for long-lasting treatment for asthma. Okay, so that's really all you need to know about these pathways. So just to kind of summarize again, if you're not looking in detail at this step, you have the arachidonic acid, omega-6. Two things you could do with it. You could either treat it with a lipoxygenase, making leukotrienes, the two double bonds, the addition of a pair of oxygens, or you could cyclize it with a pair of oxygens into molecules called prostaglandins. These are inhibited by cyclooxygenase enzymes. Both of these pathways are pro-inflammatory, which goes very far to explain, again, why having so much excess omega-6 fatty acids turns out to cause inflammation. All right, let's move on now to another completely different class of lipids. The last one we'll talk about today. And these are the lipids that you recall last semester that we talked about here, that are formed from this molecule here. These are collectively known as the isoprenoids. So you remember the isoprenoids have the isoprene skeleton, one, 2, 3, 4, 5 carbon skeleton. Remember, the position of the double bond is not necessarily going to stay in the same place. And also, remember, you cannot make any of these isoprenoid molecules at all directly out of isoprene. Instead, the precursor for all of these molecules will be isopropanol pyrophosphate. And of course, the energetics for making these will be the loss of the pyrophosphate group. So hopefully, maybe that's one of the last times I say that this semester. What are these isoprenes? Some of them are long open chain molecules. We'll see some versions of those. But the ones that we'll focus on most have the steroid nucleus here, this common four-ring steroid nucleus. And these include molecules like cholesterol, of course, as well as the steroid hormones, and the bile salts, which we were talking about when we were talking about fat digestion. So there's an example of one as well. Here is the structure of cholesterol. So of course, this does form an important component of the membrane, helping regulate the fluidity of the membrane. High cholesterol content can disrupt membrane, leading to additional fluidity. You'll recall lower cholesterol content actually helps create connections between those fatty acyl chains. So cholesterol is kind of the regulator to keep your membranes just at the right fluidity, not too rigid that they break, not too fluid that you turn into something with the consistency of olive oil. In terms of metabolically, what is cholesterol made of? Cholesterol is all acetyl-CoAs. So here are all the acetyl-CoAs that actually go into cholesterol. The synthesis is cytosolic, so not surprising that's where we're going to make it. Uses a lot of NADPH, uses a lot of ATP, 18 acetyl-CoAs. Why is that so incredibly significant? Because unlike the other fatty acid and fatty acid-derived molecules we've seen before, cholesterol is not ever going anywhere. This is pure investment of acetyl-CoA into the process of making cholesterol. The only way to get cholesterol out of your body is to excrete it. So excess cholesterol, we'll see in the last part of this lecture, will get scavenged from your body, turned into these bile salts, which will be used to emulsify fats, and then excreted out of your body in solid waste. which means you should only make cholesterol if you need to make cholesterol, right? This is 18 acetyl-CoA's work. So 18 citric acid cycle passes in addition to the excess of the ATP that's required. So it is, one of the most extreme examples of these molecules, but it's the biosynthetic pathway that we're going to follow. So once again, just like what I said before, unlike when we're talking about the fatty acid synthesis, the key here is not to try to be memorizing each individual step, but rather when presented potentially with a particular step, to understand what is actually happening. And I think the best way to do so is that before diving into the synthesis of a complex molecule like cholesterol, to kind of understand what are the different phases and what we're going to see happen. So just like we did with fatty acids, to kind of look at a little bit of a preview, if you are. So what do we have when we're looking at the actual synthesis of cholesterol? Are these 4 critical steps? The 4 critical phases, you should say, are first of all to take the acetate, right, and activate it into this molecule called mevalonate. So this is sometimes called the mevalonic acid pathway because this mevalonic acid, which is going to combine our acetates together, is going to be a crucial intermediate in the synthesis of the cholesterol. Of course, this is also not sufficiently a reactive molecule. This is not isoprene, so we need to convert this mevalonic acid into the activated isoprene. So here is our isoprene, right, that we're going to be working towards synthesizing, and here is our pyrophosphate. We're going to build that pyrophosphate in kind of a weird way. It might seem logical that nature would just donate a pyrophosphate, since they always seem to be flying around, but that's not actually how we're going to build a pyrophosphate. Once we have multiple activated isoprenes, we can connect them together into a long open chain molecule called squalene, and a series of enzymes can then carry out each of these proper cyclizations to give us cholesterol, or an organisms that don't make cholesterol-like molecules. So let's dissect this further, this connection process, this process of building. Here's isoprene, right, which, just to be clear, is not the actual component, right? Isoprene is our phosphate. But we can kind of start to imagine when we look at these molecules how we can connect them. And it turns out that there are 2 types of chemical connections geographically described that we can have in assembling these. They can be described by, I didn't invent this, but the figure of a dog. A dog has 2 heads, a head and a tail, and we can describe isoprenes in the same way. Here is the head of the isoprene, and here's the head, sorry, of the isoprene, and here's the tail. And so when we are connecting isoprene units together, we can do so by going tail to tail and make these open chains. We can go head to tail and build open linear chains, or we can make head to tail connections, and this is the modality by which we'll actually be able to get rings. So there'll be head to head connections, tail to tail connections, head to tail connections, and those head to tail connections will be, of course, involving putting the rings together. In terms of the chemistry of making these sorts of connections, well, that is something that you kind of probably recognize. You have the loss of the pyrophosphate group, leading to this positive charge here, and the attack of the electrons on another activated isoprene, actually making that carbon-carbon bond. So the chemistry itself is not necessarily particularly exciting. Okay, so let's do this. Let's get ourselves towards mevalonate. Again, not to memorize which one of the steps. Please do not go home and start drawing out every step of cholesterol synthesis, but kind of try to understand how this whole thing is actually coming together. Remember, we're going to need 3 acetyl-CoAs. Their actual assembly is not very complicated because each one of these acetyl-CoAs has a carbon atom that can act as a nucleophile, as well as a carbonyl that can act as an electrophile. Remember the localization. So by combining these together with the enzymes thiolase and the HMG-CoA synthase, we actually come up with here this HMG-CoA molecule. So this is the combination of 3 of them constantly building out by adding another acetyl-CoAs onto one of the electrophiles. You can see in this particular figure, the 3rd one here has been traced to show how this can attack here, building out the entire structure of the molecule. The final step, therefore, in the synthesis of the mevalonate involves a pair of reduction steps, right, and the loss of the CoA, and now we have mevalonate. And at this point now, we're going forward, we're going to be making some isoprene. So that should probably say the first committed step of isoprene synthesis. There is nothing else to do with mevalonate other than go going backwards. Of course, we don't need mevalonate. If we're going to make isoprenes, we're going to need to convert the mevalonate into the activated isoprenes that we actually need. And this is a really energy investment step. So we know that we're going to need to have at least 2 phosphate groups, right? We have a demand for 2 phosphate groups because that's going to form our pyrophosphate leader. Again, it might have seemed more intuitive for nature just to give us a pyrophosphate group since they're always flying off of molecules anyway, but that's not what's actually going to happen here. So the phosphate groups are actually going to be built onto the end of the mevalonate, one at a time by taking 2 ATV groups. So this is our pyrophosphate group. The third phosphate group has a really interesting use. So the third phosphate group ends up here. That phosphate group is not long for here, however, because now what's going to happen? Our same old strategy. We have a carbon dioxide here. We have a carbon dioxide here. By the time we get to this enzyme, the carbon dioxide is coming off. That's sending the electrons going this way. But for that to happen, we need a really good leaving group, and that's going to be our phosphate group. So in this case, what the phosphate group is actually doing is participating in the decarboxylation reaction by existing as a good leaving group, right? So again, we started with our mevalonate, which was just combining our 3 acetyl-CoAs together, building on a phosphate, building on a phosphate. So here's going to be the leaving group for the actual synthesis. But for now, we need to first introduce the double bonds. So here we go. We're going to do that here with the loss of the carbon dioxide. Electrons coming crashing here, sending the phosphate group right back off. We then have this first isopentyl pyrophosphate. We actually need 2 different isoprene versions to do this, but this isomerization is just going to actually happen by itself. So those molecules are equal in structure. Anyway, now things get kind of fun. Don't worry, again, you're not memorizing this, right? You're trying to understand what's happening here. We're going to start to make some molecules. And what's really kind of interesting about these molecules is that even though their fate for most of them is going to be to assemble cholesterol, we actually do see in different organisms these molecules accumulating, so they have individual functions. For example, if we take one of these dimethyl allopyrophosphates, combine it with the delta pentyl pyrophosphate, we do a head-to-tail condensation. So we have a head-to-tail condensation, lose the pyrophosphate group. We get geranyl-pyrophosphate, which seems to accumulate a lot of these beautiful geraniums. I don't think it's the cause of their color, but it does. If we do another condensation, right, in this case another head-to-condensation, losing this next pyrophosphate group, so we bring in another one of the isopentyls, do another head-to-tail condensation, we get the farnesyl pyrophosphate. Farnesyl is absolutely on the way to making cholesterol, but some of this is actually going to get siphoned off because this is actually a relatively common post-translational modification of proteins. Anyway, we're not that concerned about that. Our real goal here, of course, is to try to get ourselves to cholesterol. We make a pair of these farnesyl pyrophosphates. We do our head-to-head. This is going to give us this molecule squalene, which is found a lot in it. Right, sharks. I don't know. We're not going to sing now, I promise. We can make the semester for that. Squalene, with the appropriate oxidations, and there are far, far too many, and the additions of oxygens, is using different cyclase enzymes going to cyclize itself. You could work this out if you really have nothing else to do. Into the scleroid nucleus. Additional enzymes then will come in to provide oxidations or add double bonds. So this is where we get this whole family of related steroid nucleus molecules, is by that last step, and then local cyclization. Regulation is the last thing. So quickly before we go, key points, if you're thinking there's too much here, what do I need to know? The purposes of the ATP, right? Really significant. What each one of these ATPs are being used for. Extremely important. I think you should be able to imagine like how a mevalonate would kind of come together. Not necessarily memorizing it, but maybe given the mevalonates, find your acetyl-CoAs in there. I think that's something that's entirely reasonable to do. Really, really important, just understanding this particular chemistry, what it means head to tail. Like, I'm not going to make you make cholesterol, but sure, you can connect 2 isoprenes together, head to tail or tail to head. I would like to think so. And also, just really the most important thing here so far, and that we'll touch on when we finish the regulation, this is super energetically and metabolically phosphate. So you should know that cholesterol is made entirely of acetyl-CoAs, requires a lot of ATP, if you don't remember the number, and that's it. The only thing you can do is when you're done with the experiment, there's no cholesterol metabolism or We'll pick up and finish this next time, guys, about, I think, 15, 20 minutes or so. And then we'll do some nitrogen. This will be the end of the test. Encourage your friends to come to the last class. I don't know, it might be a crucial.